Yeah, everyone, thank you for joining our cell therapy seminar this month. We are really excited to have a very inspiring speaker that I had the pleasure to talk to recently, and this is why I found that his work is really inspiring, and I was very interested to have him joining us. And this is Peter Wang. Dr. Peter is a full professor of biomedical engineering at UC San Diego at the Institute of Engineering and Medicine. And luckily also for us, he will be joining us beginning of next year as the department chair of biomedical engineering at the USC School of Engineering starting of January 2023. So we are really very excited to have Peter part of the UC team. And today he's talking to us about the genetic engineering of remotely controllable cells for cancer immunotherapy. And I have to warn you, this is a super exciting approach. So you have to stay very closely listening to the talk, and there will be hopefully a lot of interesting questions after that. So Peter, thanks so much for joining us. We're very excited to hear your talk. It's all yours now. Thank you.
Thank you very much, Mohamed. Can you guys hear me well?
Yes, it's perfect.
Thank you. It's really a great pleasure. Although I gave another talk like 2 weeks ago, but hopefully, you will not get bored. And this talk will also provide some new thoughts and insight, and hopefully promote some collaborations between us. So today I will mainly focus on our recent work on the controllable cell for cancer immunotherapy, although I will also introduce a little bit on our other arms in the lab, for example, live cell imaging for signaling, transductions. So that's the main, picture for the lab. Mainly we have 2 arms in the lab. One, we try to engineer molecules, introduce in the cell so that we can really spy on them to understand what they are thinking whenever they are engaging with the environment or engaging with a target, cell, for example. And on the other hand, we also do, which is the bottom arm of the lab, we try to engineer molecules and control the cell using external signals so that we can guide their function to recognize specific cell and, for example, for cancer immunotherapy, kill them. So first, I would just provide a very brief introduction on our work in flat biosensor engineering to visualize molecular signals in living cells. So the concept of these technology is basically based on fluorescence proteins. Here you can see a cyan fluorescent protein represented by CFP and a yellow fluorescent protein represented by YFP. And they're connected by a phosphate amino acid binding domain and a substrate peptide. Now, you will have one molecule represent a biosensor, which is completely genetically encoded. And therefore, we can introduce into living cell. And without any activity, you will have a pretty strong energy transfer. And excitation of this molecule will allow you to see a yellow color. And if there's activity ongoing in the cell, it will cause phosphorylation of the substrate peptide. Now, this phosphorylated substrate peptide will have high affinity, binding to the phosphate amino acid binding domain, and therefore separate these CLP and YP. By the way, CLP can serve as donor, and YP is serving as the acceptor. Now, if you look at the same molecule again, you will see cyan color out. If you take out the ratio between this cyan and yellow, you will get one index. This one index will represent the kinase activity. If the index is high, kinase is high. If the index is low, the kinase activity is low. So we indeed have engineered different kind of biosensors. Here I just want to highlight one, which we can monitor SAC kinase in living cells. So this is my favorite movie. You can see we can challenge these cells with growth factor. We can see the color change. We can wash out, it will cool down, and we can challenge it again with growth factor to see the second wave for activation. And with similar kind of strategy, we can also visualize what's going on in the nucleus, what they're thinking in terms of cell activities. Particularly, we know genome are wrapping around the histone core and the chromatin, and the histone tail sticking out, governing the regulation or the genome architecture. For example, these histone tails can tell whether the cell should open or close the genome regions so that the gene will be exposed to be controlled by the transcription machinery. And we try to understand how in a single cell, these are dynamically regulated. And indeed, we designed biosensor connecting, again, a donor, CLP, acceptor, a binding domain, and of course a linker. And then we directly connect the histone tail, H3. This will allow us to insert the whole biosensor into the nucleosome, so that whenever there is epigenetic change on this chromatin structure, and for example, in this case, H3K9 is methylated, it will start to recruit the HP1 and cause a donor acceptor like getting together. And this will allow the color change of biosensor, and therefore we can track at the single cell level how this epigenetic change is controlled and how this will impact cellular functions. So we have recently, actually it's quite a couple of years now, we developed a biosensor allowing us to visualize in the nuclear the H3K9 trimethylation and monitor the cell going through the cell cycle, for example, mitosis. So this is a movie I want to play. In the beginning, you will see this nuclear, relatively yellow color or red color with a high level H3K9 trimethylation. And when they get it through the mitosis cell cycle, the epigenetic level will be reduced, and they will basically recover as soon as they finish division for the two daughter cells. So you can see in the beginning interface, relatively high, flat, and quench, and quickly recover after they finish the division. So this is my favorite movie. I will play at least twice. Interface, division, and you can see the recovery, suggesting these epigenetic signals in the nucleus are precisely controlled at the single cell level, which is not easy to be obtained if you just do a bulky like a ChIP-seq, other like cell lysis based assays. We further have developed recently a technology we can create like tens of millions different sequences for these biosensors and introduce into mammalian cell to create the mammalian cell library, and then we can use the fax machine to sort those cells with high or low flat, and then subject to a next gen sequence to identify those biosensors with higher sensitivity. And these allow us to engineer those biosensors capable of tracking the enzymatic activities in T cells. For example, we can track ZAP70 activity when a T cell engage with the surface. And you can see very nice structure and subcellularly distributed ZAP70 activity if it's mediated by TCR signaling, whereas CAR signaling are relatively less structured. We can further use these biosensors to monitor, for example, a T cell engaging with the tumor cell, what's going on at the interface. And we can see these are the signals, polarized ZAP70 activity between the CAR T cell and target tumor cell. And I probably don't have time to go through the other data. We can also use these biosensors to screen drugs. If you are interested in those kind of research and try to use biosensor to visualize more likely events in the living cell, we will be very happy to collaborate with you. But for today, I would like to switch the gear so that we can talk about some recent development in the lab, utilizing the molecular engineering to control the cell. First, I would like to introduce a concept and the project. We can integrate our sensor and actuator together to engineer so-called machinery molecules so that we can reprogram macrophage and guide them for the tumor eradication. So this I have to thank my daughters. I watched this movie together with them and was very inspired by this concept. Wally, it has a sensor and also actuator, and they can do the job whenever they detect, for example, a danger signal, right? Or based on what the environment, they can do appropriate actions. So at that time, we have played with the sensor quite a bit, but we never really create those machinery molecules. So we thought maybe we could give a try integrating a sensor and activation domain in the cell so that the sensor can keep monitoring the cellular status whenever they detect a specific signal. Of course, they will tell us, the color change, right? And then it will also open these activation domains and allow them to hook up to the activation cascade or the host cell to control the cellular functions. And we are likely familiar with one molecule and we played with it. In this case, we have this molecule called SHIP2. It has a phosphatase domain, which is an enzyme domain, but at the rest state, it's locked by its two SH2 domains. So although enzyme domain is there, but it cannot have any function in the host cell. Now, we thought maybe we can engineer this or re-engineer this molecule by adding a sensor, acceptor, donor with a linker, and a peptide containing 2,000 sites. So in the beginning, if the cell or if the molecule is at the rest, there's no flat signal. Now, if there abnormal kinase activity is ongoing in the cell, it will phosphorate these 2,000 sites. And we know phosphorated 1,000 sites will bind to the SH2 domain. And this binding will first allow the donor acceptor to get together so that you can see the flat signal. At the same time, these two phosphorated 1,000 occupy the two SH2 domain so that they can no longer hold these phosphatate domain. Now, the phosphate domain will be exposed and become functional to control cellular function. So essentially, this is one molecule, but it has two functions. One, monitoring the kinase activity using FLAT. At the same time, it can control cellular function using these phosphatase functions. So we indeed create this molecule because it's genetically encoded. We can introduce into the cell and monitor the growth factor triggered machinery molecule actions. So you can see growth factor, you can see the color change. And if we mutate the 2,000 sites in the peptide as the antenna, then you can see everything will be gone, suggesting these reactions are really based on what we designed. Now, this is a very cool molecule, right? It has sensing and actuation function, but what can we do with it? What kind of useful application we can have? So we found out there's one scenario. If there's a tumor or cancer cell, you can add antibody to recognize the tumor surface, specific antigen. And if you throw in a macrophage has Fc gamma receptor, can recognize the tail of the antibody, and therefore they will start to kill this tumor. It's called phagocytosis. Very famous in immunotherapy area. But a lot of tumor cells, they also express a very famous molecule, so-called CD47, which is also very famous, named as don't eat me signal. So these CD47 engage with another receptor on macrophage, SERPA-alpha. And these will trigger tyrosine phosphorylation and recruit negative regulators to shut down macrophage. So essentially, you can imagine the macrophage is a car. It has an engine guided by antibody, Fc gamma receptor to try to kill the tumor, but it also has a brake, which is SERPA-alpha, which can be turned on by CD47 and shut down. So we think maybe we can re-engineer this brake receptor so that this brake will no longer be stopped by CD47. It will actually help the macrophage to keep eating if this CD47 is presenting by the tumor cell. So we engineered this chimeric molecule with SERPA-alpha brake receptor on surface, but we have this peptide, which can be tyrosine phosphorated, triggered by the SERPA-alpha. And whenever the tumor cell using CD47 and turn on the phosphorylation, it will recruit the engineered binding domain or the SH2 and open this phosphatate domain we engineered to help the macrophage. So this is a movie, basically, you can see on top, we are basically having successfully engineered the macrophage, and you can see the color. And we are going to drop antibody-coated tumor cell expressing high level CD47 on top. you will see one tumor cell will come, you can see the flat change, you can quickly be killed and cleared. On the bottom, we purposely delete the enzyme domain we engineered so that these engineered macrophage can only see what's going on using flash signal, but they cannot help to eat. So you can compare. A lot of tumor cell will be dropped, but they will be like dragging around on the surface. You can see they're approaching, engaging, but they can never be eaten. That's why they stay there, never be clear out. And with this similar approach, you can even use this engineered macrophage or monocyte introduced into tumor cell. And these are the group. We introduce this engineered monocyte and macrophage. And you can see compared to the controls, we can suppress tumor significantly. You can also see these are the control groups, and these are the just naive monocyte, and these are engineered monocyte. We can suppress tumor strongly. So I hope with this result present evidence to convince you that we can indeed create those smart molecules. They can cruise inside intracellular space and keep monitoring the status in the cell. And whenever there's a correct information comes, they will fire and help the cell do the correct actions. And we successfully show that indeed this works in macrophage. Now we can re-engineer this macrophage so that they can kill those CD47 high tumor cell, which was typically not killable by the normal macrophage. So we consider this engineering as a a molecular version of the Tesla car or Google car. Basically, they are all automated. Very nicely, they can control everything by all themselves. So this is pretty nice. The only problem is if there's anything wrong in the middle, what can we do with it? We can't, right? Because everything is automated. So we start to think maybe we can also engineer molecules so that we can control from outside, again, using engineering approach. So this basically I would like to introduce or also relatively recent work using light or ultrasound to control the CAR T cells to kill the tumor. So CAR T, many of us probably are familiar. It has this so-called chimeric molecule with extracellular domain, recognizing the tumor antigen, and also have activation domain. So if you engineer a T cell expressing this CAR molecule, when you put back into patient, this CAR with the antibody recognizing the antigen, bring the T cell next to the tumor cell. At the same time, the activation domain will be turned on so that they can secrete like granzyme and other cytokines to kill the tumor. So this is a concept of the CAR T, and we all know that there are a lot of success, particularly for blood cancers, right? So Emma Whitehead is first, to be rescued from advanced leukemia by Carl Jung in Children's Hospital of Philadelphia in 2012, and it has been 10 years now. A very successful, example. And actually, there are also a lot of successful, cases for different blood cancers. Now, there is indeed, still a big problem for solid tumors, particularly for solid tumor. One, you have like a cytokine release syndrome. Another, I think it's more severe, so-called on-target off-tumor toxicity. So in this case, because solid tumor, a lot of times they are actually originated from the normal tissue or organ, right? So they express similar level of profile on the surface as tumor cell. So it's almost impossible to identify a tumor-specific antigen only, expressed on the tumor cells, but not expressing in other normal organ or tissue. So these present the difficulty because if you imagine, if you introduce the CAR T into patient, they will go everywhere. They will, of course, attack the tumor, kill them, but they will also cause damage for some normal organ when they express even minor level of this antigen. So that's so-called on-target off-tumor toxicity. And because the human physiology is so complicated, it's almost impossible to really, no matter how you optimize this antigen antibody pair, it's just almost impossible to find the perfect one. So we are thinking, can we change our thoughts to basically design a strategy so that we can engineer these CAR T cells? They can be controllable, and we can control them so that they can only be activated at a tumor. They can go everywhere, no problem, but they will be only activated when the external signal shining on this tumor region. So therefore, we can avoid the on target of tumor problem. Even this antibody or CAR is not perfect. It's fine because they will not have toxicity in other places. So that's all thought, and we try to demonstrate whether this thought is achievable. We first did work on light controllable CAR T cells. So in this scenario, we engineered these three guys. One is inducible promoter driving a CAR or a reporter. And we have another one, which has DNA binding domain with a light sensitive domain together with a light switchable nuclear localization motif. So this guy will be kept outside the nuclear all the time. And we have another one, which is also light sensitive Cry2 connect to a transcription activator. Now you have three players. These two will be mostly inside the nuclear, and the other one will be outside the nuclear, so they don't have the chance to interact with each other. No activity will be observed. Now, if you shine the light onto the cell, this BINLS will flip. Basically, the nuclear localization motif will be popped open and exposed. And this will lead the DNA binding domain and CRB1 to go to nucleus. And CRB1 and the Cry2 can dimerize, and the net result will be these three guys will be homing at the same place, and the inducible promoter can now be turned on by the transcription activator to produce the reporter or CAR. So essentially, you can using these genetic circuits to convert a light signal into a gene production. We first demonstrate indeed this concept works. We can use light to push those molecules in and out of the nuclear. We can turn on the light, it will go to nuclear. You can see the nuclear become brighter, and they can quickly be pushed out if we stop the light. So we can flip whenever we want them to go have access to the inducible promoter gene in the nuclear. And this, of course, can be translated into the gene production. So before light, there's no gene production. And after light, you can see the gene can be turned on. So these genetic circuits indeed can allow us to convert the light signal into gene production. Now, whether we can use it to create light controllable CAR T, and we indeed did that in mice. So in this case, we create a light control box so that we can control the light pattern precisely. Like every 30 seconds, we only activate for 1 second. And we can use a blue LED patch to the tumor site or the mice, and we can use a Johnson & Johnson patch to do that. It's quite convenient. And then we can use this light control box to turn on the genes in the particular location. And these are results on the right. As you can see, if we don't shine the light onto them, these are the area where we inject the light controllable cells, no gene production will be turned on. But if we turn on the light, this is before, this is after, we can see the gene production is, pretty significant. And these are quantified results we can turn on like 20, 30 folds comparing to the control upon the light activation. Now people ask whether we can use these to control the CAR T function and suppress tumor. So we did this experiment. We introduced tumor cells expressing luciferase and monitor their growth. Of course, they grow pretty well at 16 days. It's quite big already. Now we introduce another group. We introduce tumor cells expressing luciferase and introduce light controllable CAR T, but we keep everything in the dark. We don't activate these CAR T cells. You can see the tumor still grow pretty well. Now for the last group, we introduce the tumor cells and then introduce the light controllable CAR T. And in this case, we activate them with light shining. And you can see the light indeed activate CAR T and suppress tumor. one mice may be feeling itchy and bite the cable and run away. So we lost one data, but the result was still, impressive. It's actually beyond our expectation. Suppression power comparing to the controls. So that's quite nice. And particularly this will be very useful for any like melanoma or tumor expressed on the surface. But what about deep tissues? We know light, they cannot penetrate deep into tissue, right? So we are thinking whether we can engineer other modality which will allow us to reach deep in the animal. So we consider ultrasound. Ultrasound, we know it's a mechanical wave. It can penetrate very deep in the soft tissue. And if you can focus the energy, you can really, create a strong, force inside a particular location. There are quite a lot of expertise at the USC, so I'm very, happy, and we'll have the opportunity to collaborate with them. And we first actually, this is also a collaboration work with faculty at the USC, Kirk Schoen, professor in biomedical engineering. So we basically integrate ultrasound and our imaging power together so that we can simultaneously using ultrasound to control cell and monitor the cellular activity in these living cells to test whether these two modalities can be indeed integrated together. So from the bottom, we can use fluorescence flat to monitor the cell activity. On top, we can use ultrasound to activate these cells. In that case, we utilize one microbubble because we want to amplify signal. That was like very early, nobody really have too much expertise on that. We were probably one of the very first try to integrate these two things together. So we try to amplify signal first using microbubble because we know microbubble, whenever ultrasound pressure comes, it will shrink, right? And when the ultrasound pressure goes, it will expand. So essentially, ultrasound pressure or wave can cause this microbubble to oscillate in size. So this will provide a very neat system for us. We can coat the microbubble with like a streptoavidin, and then they can bind to a biotin, to the antibody. And if the antibody is connected to a mechanosensory channel, piezo-1, expressed on the cell, you will have this sensing and amplification system. So ultrasound remote signal can reach the microbubble, and microbubble can mechanically vibrate, right? And this mechanical vibration can be translated into the piezo channel. And we know inside cell, it's only like 100 nanomolar calcium in concentration, and outside, typically it's millimolar. So this mechanical signal opening the piezo can allow the calcium from outside to flush into the intracellular space of the cell. And we demonstrate using our integrated system. Indeed, when we introduce calcium bound sensor into the cell, 10-second ultrasound can already trigger very nice calcium, influx into the cell. And the 40-second, it quiet down. And this can be monitored by the fluorescence microscope. And we can also show the system is very specific. If you take off the microbubble, for example, here, or if you chelate the extracellular, calcium, or if you remove the piezo-1, right? In this case, all the signal will be drastically suppressed, suggesting this ultrasound triggered calcium signal is precisely, following what we designed. So calcium is very nice and it can be controlled by ultrasound, but that's not what we want. We want to control gene so that we can eventually control the cell function. So we further tested this concept, whether this ultrasound controllable calcium can control gene activities. So we know calcium can activate calcineurin, which is a phosphatase. And phosphatase can dephosphorate a transcription activator, NFAT. NFAT typically stay in the cytosol, and when it's dephosphorated, it will go to nucleus to activate the gene. So if we can engineer a synthetic gene cassette with an inducible promoter receiving the NFAT signal to drive the gene, then can indeed convert the calcium signal into a gene production. So basically, this whole design should allow us to convert this remote ultrasound signal into calcium signaling and the calcineurin NFAT eventually designed gene production. It could be a car. So we first demonstrated this concept indeed works. So as you can see, we introduce this piezo one into these, hex cells. The red color represent engineered cells. Without ultrasound, this reporter GLP cannot be produced. Now with ultrasound shining on these cells, the engineered cell indeed can produce the final product, the GLP. And we also have demonstrated, this GLP of course can be replaced by CAR gene so that ultrasound can also control the CAR gene production. But then there's another issue which probably will prevent us, to apply this technology in vivo, mainly because we need to utilize a macrobubble to amplify the mechanical vibration. In vivo, it will be much tougher for us to do that. So we thought, can we completely remove this macrobubble and still use ultrasound to control the gene activity in cells? And we learned that in clinics, actually, indeed, there are approaches people use it, so-called the high intensity focused ultrasound. So essentially the concept is they can use multiple transducer ultrasound and focus the beams onto a particular location to raise the temperature for the tissue. And people indeed use that kind of technology, to, for example, for prostate cancer, they can use this temperature to burn the tissue and therefore kill tumor. Of course, that's not a very, I would say, precise usage of the ultrasound, because you raise temperature to such high degree to burn the tissue. Of course, tumor will be burned, but it will also create a lot of damage on these neighboring surrounding tissue, right? So we thought if we can integrate with the gene expression circuits, then we can probably make use of this ultrasound much better. So we try to test this concept, whether we can use ultrasound to control temperature and control the gene. And in this case, we do not need to raise temperature to such high degree anymore. We can only use like 43 degree, which is lightly warm, mild temperature increase, and see whether we can turn on the gene expression. So we introduce this so-called heat shock promoter driving the gene, and we can indeed see with ultrasound to focus and raise the temperature only to 43 degrees, we can see the gene expression very nicely. As you can see, this is a control without ultrasound and temperature raise, gene expression is almost none. With this activation, we can see the gene production. Even better, because these ultrasound are precisely controllable, so that we can, create different kind of patterns. In this case, we can even create, for example, short paused ultrasound waves, like only 5 minutes, and break them, repeat 3 times or 2 times. You can see the gene expression is almost the same as if you, continuously raise the temperature to turn on the gene. So this is particularly useful because long-term temperature raise perhaps will cause some damage to the T cells, which is quite delicate. Now, if we can break them into short pause, then the cell can survive much better, and we can still control the gene very well. And then we further tested whether this concept indeed can allow us to control the CAR expression. So we designed this gene cassette. One is heat shock promoter driving a CAR connect to EGFP so that we can see the green color. Another one, we use PGK, which is a constitutive promoter driving mCherry, which is a red color, so that we can know which cell is successfully genetically engineered. So we did this experiment. You can see without stimulation, this engineered cell, which can be monitored by mCherry, no GLP is produced. Now, if you turn on the ultrasound and temperature increase, you can see these engineered cells, they can produce CAR nicely on the surface. That's why you can see this ring structure on the single cell level. So these are, very encouraging, and we were very excited. These are all, very successful, but mostly in the in vitro culture dishes. We also want to test whether this works in vivo. So we create this system integrating focused ultrasound which can allow us to guide the beam and focus on particular location in the mice. But we connected to a MRI system so that MRI can keep monitoring the temperature of these mice and provide a feedback to control the power of this ultrasound so that these temperature can be precisely controlled. For example, if we want to create short pulse, 43-degree waves, this can be done by this integrated system, but cannot be done by only ultrasound because the temperature monitoring and feedback would be critical for us to have a precision of the temperature control. So with this system integrated, I just want to show you the configuration. This is the top view of the ultrasound array, and this is side view of the ultrasound. You can see this focused point can be guided X, Y, or Z direction. Essentially you can guide this focused point to any place in a mice. And the MRI can allow us to keep monitoring the temperature profile in the mice. So this is the profile. And this can provide the feedback for the ultrasound power so that we can maintain the temperature profile precisely. Now, if we introduce these ultrasound controllable cells in the mice, and if we don't have ultrasound stimulation, no gene production, and 10 minutes continuously, of course, gene production, and short pause the wave, 5 minutes, only repeat twice, we can also see very good gene production, suggesting the whole system indeed work in vivo. Now, we want to test the final task, whether we can use these ultrasound controllable CAR-T to engage and kill tumor. So in this setting, we try to do this experiment. We introduce tumor cells on both sides of mice, in the same mice, left and right. And then we introduce ultrasound controllable CAR-T on both sides. But we only activate on the left, leaving the right side as a control, and compare whether ultrasound can indeed activate this ultrasound controllable CAR-T and suppress tumor on the left. So this is the result. Just with one dose of the focused ultrasound, we can clearly see the suppression power. These are the left side, this is the right side as a control. And these are images. Remember, it's exactly the same mice, left, right, for all these 4 mice. And you can see in the beginning, they are similar in tumor size, but as time goes on, at day 18, we can already see ultrasound treated group versus the control group. Again, these are the same mice, we are just comparing head to head. And we can see a clear suppression for the ultrasound controllable CAR-T. Now we call this CAR as fast CAR, focused ultrasound controllable CAR. And these are basically, we are using this lymphoma tumor model as the demonstration for the technology. We also tested in the real solid tumor, which is prostate cancer. So again, the design was similar. We introduced prostate cancer on both sides. Then we introduced ultrasound controllable CAR-T on both sides, and then only activate the left side. And the result was quite similar to this lymphoma tumor model. And again, same mice, and you can see the left and right, we can see drastic tumor size difference. And we imagine if we do multiple dose of this ultrasound treatment, the result will be even more significant. Just like we can do a massage every 2 or 3 days for the subject, and we perhaps can achieve even stronger tumor suppression. Then people start to ask, then how this will be better than the standard CAR-T? And we did this experiment. We basically again introduced 2 tumor sites with same antigen expression, and then we introduce a standard CAR into only one site and monitor the tumor size. And you can see, we call this CAR-T injector site as proximal site, this as the distal site. Sorry, this is the distal site. This tumor CAR-T injector site is the proximal site. And you can see proximal side, of course, the CAR-T directly, kill them, and these tumor never grow up. On the distal side, the tumor grow up, but eventually also being suppressed and killed. So this is an experiment to mimic if there is normal organ tissue expressing similar antigen on the distal side, they will also be attacked and killed. So that's a major problem. Now, if you look at our FAST-CAR, again, we introduce two tumor on the sides and only introduce FAST-CAR on the left side and activate them. And you can see the proximal side, ultrasound activation, kill the tumor. But the distal side, the tumor grow pretty well. Suggesting if this is a normal tissue organ expressing some similar antigen, they will be fine and spared. So these results clearly suggest all FAST-CAR have much better specificity and precision, and therefore less on target of tumor toxicity. So this work published very recently in Nature Biomedical Engineering. We were very lucky, they select our work as the cover for that issue. So we basically illustrate that we can use remote ultrasound to activate gene in the CAR T and therefore kill the tumor only on the tumor side. So I hope I presented enough evidence to show you that indeed we can, engineer the cells so that we can have the sensor, actuator, and we can control them precisely. We are also thinking, hopefully in the future, AI or deep learning can help us to connect the whole loop together so that we can optimize, the power and the pattern of the treatment so that we can achieve optimal outcome. And that's our dream, hopefully in the future, we can have patients with this jacket with different transducer. Now we can use cellular phone to control the program of these ultrasound transducer functions and therefore control the tumor or even other disease as well. So I'm a bigger fan of this, clinical center building at the NIH. They really, have the clinician and the researchers, scientists, engineers all working together, close the loop using cutting edge technology to directly apply to the patients. And we are really hoping all, USC can have similar kind of organization. We can collaborate all together to form a team so that engineering school, cutting edge technology, and the medical practice and the hospital, activities can all be, coordinated, and we can really, shorten the distance and close the loop and form these, elevated the level, directly applying the cutting edge technology to the patients. That's our dream, and would really love to work together with everyone here toward this, ultimate goal. I have to thank my lab members, particularly Dr. Mingxin Ouyang, Dr. Longwei, and Mint, and Dr. Qingpeng for the biosensor development, and Dr. Jason and Lei for the engineered macrophage, and Dr. Ziliang Huang and Molly Allen for the light controllable CAR-T, and Dr. Yijia Pan and Dr. Shelley Wu for the ultrasound controllable CAR-T. And we have a lot of collaborators. I'm always, grateful. And we are also, grateful to the funding support as well as UCC for all this work. We'll be happy to answer your questions. I think that's my last slide. Thank you, everyone.
Thanks so much, Peter. This has been very enjoyable for the second time for me because I went through this with you, and it's really exciting, the technology that you built. And I can tell you that it's not a dream anymore, actually, what you were talking about. Actually, it's a reality that we have now, strengthening the infrastructure to translate these technologies to the clinic. Now having the new GMP facilities that we're building at USC. So once you start with us next year, I think we would immediately go ahead and plan things to bring this to the clinic and through GMP manufacturing and IND development for a clinical trial. So we definitely are excited to have you joining us next year and to push this project and collaboration forward.
Yeah, that's going to be exciting. I also really, look forward to hopefully our technology can be, directly, integrated with your facility and expertise and applied to the patients. for
Absolutely.
clinical trials. Mhm.
Yeah, absolutely. And I'm sure everyone else shares the excitement and potential collaboration with you to reach out in case they think that, they can also utilize this technology via collaboration with your team. I know we have a few minutes, so we can ask some questions. Maybe I can start with this one, and I think we had this discussion before, but maybe I repeat it so all our attendees as well, like get familiar with this point, which is like, so basically what you described to us now, when we look at the ultrasound controllable CAR T cells, is that you might require continuous exposure of ultrasound in order to keep the CAR T cell activated. And we did talk about that, and maybe you can comment on this, like how feasible do you think this would be in clinical settings? And do you think like, how would you be able to control any kind of like adverse events or hyperthermia that might happen to the patients if we expose them to a long-term ultrasound?
Outstanding question. Thank you very much, Mohamed. So I think our system, in my opinion, currently is very close to the clinical setting, because the promoter, the heat shock promoter, is endogenous. So this is directly pluggable into the host T cells, right? And the CAR gene has been well tested in clinical trials, in multiple clinical trials. So they are already being tested in terms of its safety and medical compatibility. So I think this definitely would be directly applicable for clinical trials. And in terms of the duration of the ultrasound, that's also the advantage, because we no longer need to have too much power. We only need to mildly increase the temperature for a short period, for example, 5 minutes, right? So maybe repeat one time. So these drastically reduce any side or toxicity on the T cells. Actually, I did not have the time to show these, supporting results. Actually, we indeed compared to T cell, primary T cell, head to head to the engineered T cell, which are exposed to the ultrasound stimulation. We did not see any like a difference in terms of viability or toxicity, suggesting the pattern we choose with the relatively low temperature and the short duration ultrasound treatment, they are completely compatible to control the CAR T.
And can you comment on the durability of activity of the CAR T cells themselves? Like, do you think you will need another exposure by ultrasound in order to continue the cells be activated in the tumor site?
That's another outstanding question. So you probably also aware, Crystal McCall in Stanford, right? They basically demonstrate with this kind of reversible activity, perhaps it's much better than a continuously active CAR T, because people are really recognize in solid tumor treatment, if we have a continuously active CAR T, they got quickly exhausted, which is a major problem. Now, if you can add, like in Crystal McCall's case, she added the inhibitor to allow these T cells to stop functioning for a certain time, and then remove the inhibitor and allow them to be activated again. And these allow the T cell to have some break and rest, and they become more persistent to kill tumor with higher efficiency. The same scenario will apply to all cases, but we probably don't need any additional inhibitor or anything. We just turn on the CAR T with ultrasound for like 1 day or two, and then it will naturally rest for a certain time, because we not driving them continuously, right? Now, after 2 days later, we do another massage using ultrasound. They will be activated again, and they will do another wave of the treatment. So this will be actually naturally to create some kind of waved treatment. And this will allow the T cell to have rest in the middle and have more persistent effect on the tumor. So I think for us, probably even better, we no longer need any inhibitor to be added in the middle to suppress the T cells. Usually, a lot of inhibitors, you probably know, they're not that clean, always cause other side effects. You never know. For us, it's all natural. Just relatively low temperature increase in the local region. So you feel a little bit warm in the local area. And then this will be paused for different time duration as we wish.
This is exactly the point. I think you, as you mentioned, that you actually give a chance to the T cells to rest, and you have a sort of like a wave-like treatment approach where you can expose the tumor site to the activated CAR T cells, but in a modular kind of way. Actually, Nadia was asking the same question that I was just discussing with you now, like what is really the long-term outcome, what happens to these engineered cells when they are no longer activated. And I think you did answer this now. And so basically they stay in the tumor site, they are not activated, so they will not cause any harm or toxicity unless they are exposed to heat. What if the maybe just came to my mind, what if the patient has fever suddenly? Do you think this CAR T cell can get activated?
So yeah, of course, the temperature, right? But 43-degree is quite rare, right? If really needed, of course, we can also integrate with some, well-tested drug. So that only drug and temperature together will trigger precise activation. That's relatively easy to do. We have some practice like tamoxifen, those kind of combination would be achievable. But I think for now, perhaps we can use these current technology should be quite safe. Most of fever, I think, probably cannot reach 43-degree. Most of them 42-degree or 41-degree, right? Typically.
Yeah. Yeah, exactly. Thank you. So we have a question from Martin and then Shihab. So Martin is asking like mostly about the biodistribution of these cells. Have you looked into the biodistribution and how the cells actually behave after infusion than just subcutaneous? So how did you track them for the fate of the cells or the biodistribution?
Yeah, thank you very much. That's another outstanding question. We have not really done this orthotopic, solid tumor model yet. That's actually one of the reasons. Also, would love to collaborate with folks there, to push toward this direction. For our current model, we are using prostate cancer. It's very difficult to create a orthotopic, prostate in mice. Actually, at least we did not have any, experience at that. But would love to collaborate with folks there and push this orthotopic solid tumor, direction.
Perfect.
I think I can also answer, Nadia, about the CAR T lifetime, right? Like, for example, each pulse or the ultrasound to turn on CAR, how long they can last for being active, right? Typically, the CAR protein degradation time is about 12 to 24 hours. So one dose or ultrasound can trigger the CAR T cell to be active for this period. But we are also designing new gene cassettes so that they can be longer, for example, 48 hours. These are all doable with additional cofactor.
Shahab, go ahead.
Peter, great talk. Again, my question again relates to some of these things. As you said, in SynNotch technologies and gated technologies, the protein degradation is around 8 to 12 hours, while when you make protein, it takes longer to make the protein, right? Just a simple biology of protein making. So when you give these pulses, have you done the experiments where you exactly measure whether you actually do a long-term or in the pulse that changes the amount of protein made? And that relates to my second question, which is you're doing a sub-Q injection of your tumor's T cells into the tumor currently. I could see a very interesting sort of like usage for these high ultrasound frequency use, which in medicine right now, they're used for like treating Parkinson's disease. They've actually treated in some of the solid tumors, for example, in the uterine tumors where they're like getting too big, or smaller tumors. And we could see that you could inject the T cells into these tumors, but what is the effector to target ratios required to inject that would actually, when you turn it on, to be able to stay and let's say for a couple of days, control the tumor? So one question, I guess I haven't read your paper, but how many T cells do you actually inject? And do you know how many tumor cells there are in your solid tumors when you inject them to control?
Yes, yeah, outstanding question. First of all, let me answer the question on the lifetime of this CAR, right? So I think that probably depends on what kind of domain you connect to the intracellular tail. For example, we definitely know if we introduce a GFP at the tail of the CAR, it significantly elongate the lifetime of this CAR molecule on the surface. Of course, you can try different stabilization domains, and these perhaps can be tunable depending on what you want to make it short or long. These are all doable. That's one. And in terms of the localized, resident time for this CAR T, that's definitely a question we are actually not fully, clear yet how long they can hang around. But sounds like after these CAR T activated by ultrasound, maybe there is a transition in their, status. Activated CAR T seem to be more like less mobile to move out. But that, quantitatively, how much we don't have information yet. That's something which we want to study. We think activated the CAR T because the transcription started, all this machinery being utilized. Therefore, they somehow, switch the modality from the migrative state so that they stay actually hang around in the local region longer than non-activated CAR T cells. But of course, we can also engineer a second receptor to, hook up to a local, like for example, antigen to stay even longer in the tumor region. But this research, we probably need to have more effort to dig out what's the kinetics, and the distribution. We don't have those information yet.
But the current example that you showed that you could control the tumor on one side and not in the distal side, how many T cells to tumor ratios roughly were injected?
Oh, outstanding question.
Like if you put 100,000 T cells, will it still control the tumor versus you put 10 million T cells?
Right? Yeah, but at least we can see the contrast, right?
So no, I agree. No, I'm just curious as what is the minimum required T cell to tumor ratio,
right?
That would control those types of tumors.
Right. We did not quantify the the cell number of the tumor and the CAR T when we inject them. We only know when we inject the tumor cell, it's like 200K, but we use 1 million CAR T to inject. We don't know actually how many tumor cells after this. I think we have two models, 4 days or 7 days. So after 4 days or 7 days of growth from 200K or tumor, what's the cell number? We don't have a clear, But I guess probably they will reach million something. So 1-to-1 ratio, roughly.
Great, thanks.
Okay, perfect. I think we were close to time, and I don't know if there are any questions, but I don't see any in the chat. So I think that's it. And I would like to thank you again, Peter, for joining us today and for this interesting talk. And again, looking forward to working with you soon when you join us here. Thank you.
Thank you very much. Really appreciate everyone.
Thanks. Take care. Thank you.
Okay, bye.
Bye-bye.